All right, it's my pleasure to introduce Dr. Ryan Summers. He's a Reichhold Shoemaker Assistant Professor in the Department of Chemical and Biological Engineering at the University of Alabama. He earned a bachelor's in biological engineering at Utah State University and a PhD in chemical and biochemical engineering at the University of Iowa with Dr. Mani Subramanian, followed by a postdoc research at the Center for Biocatalysis and Bioprocessing. Dr. Summers joined the University of Alabama in 2014. He's been a counselor at large for the Institute of Biological Engineering and is currently advisor for the Alabama IGEM team. His research involves metabolic engineering and synthetic biology for biocatalytic production of chemicals. I will mention as well that he has been the bishop of his local ward for the last 2.5 years. He's 39 years old, so it was kind of been interesting, being an assistant professor, but also a bishop at the same time. And this year, 4 hurricanes have come through the area. So on the latest one, I think Sally, they've been helping with some of the cleanup effort. So we really appreciate his academic contributions, but also his other contributions as well. And we look forward to hearing from him today. He is going to be talking about some of his research on metabolic engineering of microbes for high production of high value biochemicals. So with that, I'll go ahead and turn it over to Dr. Summers. So thanks.
Thank you, John. I appreciate that. And as I'm going through this, feel free to just interrupt me if you have any questions, either shout them out or in the chat. I'll try and keep the chat window open here. I'm really excited to be here talking with you guys today. I had the opportunity to give a seminar at BYU about 8 years ago when I was a postdoc. Really enjoyed that visit. Since then, I've joined the faculty down here at Alabama about 6 years ago or so. So I'd like to kind of first off start talking, if I can get this going now. There we go. A little bit about what we have going on here at the University of Alabama. So we, in the Department of Chemical and Biological Engineering, we have 23 full-time professors and instructors. One of those is in the dean's office. One of those is actually the vice president for research. So we don't really ever see the two of them. Of the 21 full-time professors or teaching faculty, we have 11 of us are assistant professors. So we're a fairly young department as well, with a lot of growth. And a lot of that growth you can see actually in this graph as I've plotted the undergraduate student population over the years here in the blue. And you can see about 10 years ago, that undergraduate population really took off from where it was about 200 per year to now, with the slight dip, we're at about, last year we were at 836, and this year we're about the same number. Graduate program's been fairly small. That's here on the right, and in the red. And so we've seen some increase recently with all of the new assistant professors coming in, and with some good funding we've been able to get over the last few years. And I believe we're up to closer to 45 this year. So we're finally seeing some growth in our graduate program as well. In 2019, we awarded 206 bachelor degrees in chemical engineering, which ranked 5th in the country. So we're pretty excited about that. In terms of the research that goes on here, so there's research in kind of some typical or traditional chemical engineering disciplines like catalysis, electrochemical systems, molecular simulations. You've got carbon dioxide sequestration. We got a lot of water quality research and membrane separations. On the biological side, which is where I sit, most of my work's metabolic engineering and synthetic biology. We have some cancer research as well and some biomedical materials. Now we are housed here in the Shelby Quad on the University of Alabama. It's fairly new, and so these buildings right here make up the Shelby Quad, and they've spread us out based on research. So we don't have the whole department together, which can be good. It can be some challenging sometimes as well. But the biological engineering group is kind of right here in this building. It's called the Science and Engineering Complex. We shorten it for SEC. It doesn't stand for the SEC conference, but we do have the SEC building right here, and chemistry is right here in this pentagon, and then the biology department is right here on this end of the building. So we're sandwiched right between the two, and that's been really good for us in the biological engineering side of things. It just helps us to have those connections right next to us. I would probably not get tenure when I go out for it if I don't mention the fact that we also happen to have a football team associated with the university. They're pretty good. Although everyone's kind of walking around on eggshells right now, just with the head coach's recent COVID diagnosis and number 3 Georgia coming into town. I also just want to point out real quickly, we do have 2 REU programs here in the chemical engineering department. So there's one that's on advanced polymers. That's going to be in its 3rd year. This has been a very successful program. They've renewed it a few times. And then we have a second one that Matt Thompson and I just were awarded a little bit ago. This is on interdisciplinary biotechnology. So Matt Thompson's in the chemistry department. And so this brings in faculty from chemistry, biology, and chemical and biological engineering to look at biotechnology research. So if you happen to know any undergraduate students that are interested in research, please feel free to send them my way in terms of contact info. I'd be happy to talk with them about any REU programs they might be interested in. I know we've had some REU students from BYU in the past that have, one of them is now out here just finished his PhD here. And then we have another student that he didn't do an REU, but he's out here as a graduate student in the metallurgical engineering department. And in the small world that it is, he actually did his research there at BYU as an undergrad, working for one of my former mission companions, Dr. Eric Homer. And then we also have GAN fellowships for new grad students. So if you know of any undergrads that are interested in graduate school as well, I'd be happy to talk with them. Just please send them my way. Okay, so Tuscaloosa, and this is my last kind of intro slide of everything that's going on here. Tuscaloosa, you can see we're sitting right here, kind of west central Alabama. So we're about 4 to 5 hours from the beach. I'll be going down to do some more hurricane cleanup this weekend. It's a beautiful area, lots of trees. We do have, and this slide I show only because we're here at BYU. We do have a stake that's centered here in Tuscaloosa. It is it was basically formed about a year ago. We have 2 family wards here in Tuscaloosa itself, and then a YSA branch as well. And the temple is right up here in Birmingham. So it's about an hour drive away, and we're lucky to have it so close to us. And then finally, our claim to fame, at least in the church, is Elder Johnson, Peter Johnson, who spoke about a year ago in General Conference, was our stake president. In fact, he's the one that extended the call to me to serve as bishop, and that was the week before he got released as stake president. So I'll stop right here and focus more on the research now. My research, I mentioned it's biological engineering. We focus on metabolic engineering, synthetic biology, and biocatalysis. So as we're looking at this, the metabolic engineering side, we have characterization of enzymes and genes, pathway engineering, where we're actually trying to engineer new pathways or transport pathways from one organism to another, and then balancing all the redox reactions that go on inside the cell. On the synthetic biology side of things, we have transcriptional terminators, origins of replication, promoters, just trying to characterize all of these things for different types of cells. We've been developing some modular plasmids, which I'm really excited about, and if I have some time, I'll touch on that very briefly at the end. And then we also have On the biocatalysis side, we've got resting cell assays where we're testing the cells that we construct. We're making these whole cell biocatalysts that we can use to carry out the reactions. And ultimately, we're mostly focused on producing these high-value biochemicals. So we mostly work with microbes. So I don't really do much with mammalian cells, plant cells, insect cells. We focus more on the bacteria and on the yeast cells. So we do a lot of work with E. coli, a lot of work with Pseudomonas putida, which is a soil bacteria. A little bit of work with Lactobacillus plantarum, that's a probiotic bacteria. And then we're just starting up some research now with Halomonas, which is a saltwater bacteria. On the yeast side of things, we do a lot with Saccharomyces, which is your typical baker's. Pichia pastoris is another common one that we use quite a bit. And then Euroia is an oleaginous yeast that we've just started working with. Okay, I'm going to focus mostly on this class of compounds called methylxanthines today. Caffeine is the most famous methylxanthine that's out there. You'll see it's a purine ring structure with 3 methyl groups. There's a methyl group at the N1 position, the N3, and then the 7 nitrogen as well. Theophylline is another one that's used quite a bit, and you see methyl groups at the 1 and 3 nitrogens, but the N7 does not have a methyl group attached to it. And there's several other variants of these. These are natural products, but there's also several synthetic derivatives. You know, we most often think of them in coffee, tea, chocolate, soft drinks. I've heard they're even selling caffeinated drinks at BYU now, so I know some people are fairly happy about that. Pharmaceutical components as well. These are used quite often as stimulants or as diuretics. Theophylline has traditionally been used as a bronchodilator for asthma treatment. Now there's some more synthetic derivatives that are used that are a little bit better than theophylline just by itself. They can also be used in cosmetic components, and riboswitches are just these small RNA fragments that will actually selectively bind to ligands. And so you can use these to control gene expression based on the presence of the ligand. And so there are several theophylline riboswitches that are out there. And they'll discriminate against caffeine by a factor of 10 to the 4th or 10 to the 5th. So you can add some caffeine to your culture and it's not going to do anything. But you can add theophylline and that's going to turn the genes on or off based on how that riboswitch is set up. So some really cool things that you can do with these methylxanthines. Most of what I'm going to talk about today is actually producing some of these methylxanthines. So we have a bacteria called Pseudomonas putida CBB5. And actually this slide I'm going to show right here is almost my entire dissertation from my PhD, which is kind of depressing sometimes when you think about my whole dissertation can fit on about 1 slide. And I spent all that time doing it, right? But this kind of summarizes everything where we're looking at the metabolism of caffeine by this bacteria. And what we see is the bacteria really likes to take caffeine and it works on this N1 methyl group here and pulls that off to form theobromine. So theobromine is the main constituent of chocolates. That the N3 methyl group is then removed to form 7-methylxanthine. And then the 7 methyl groups removed to form xanthine. Xanthine's oxidized to uric acid, and the xanthine and uric acid, these are common to all forms of life, and so they're able to break these down really easily. You'll see that I've kind of color-coded these arrows as well, where we have ndmA and ndmD carrying out the N1 demethylation. So when we remove the 1 methyl group, it's a red arrow, and that's carried out by ndmA working in tandem with ndmD. The blue arrows are showing the removal of the N3 methyl group, and that's carried out by ndmB that's also working with ndmD. So you have to have both A and D for the first one. You have to have both B and D for the second one. And then we actually have to have ndmC, D, and E together for the third step. And then finally, these dashed arrows down here represent a minor pathway. So about 2 to 3% of the caffeine goes through this lower pathway, where most of it follows through the upper pathway. I'm just going to highlight right here this paraxanthine compound because I'm going to come back to this in a little while. So don't forget we have this minor pathway, but for the first little bit, we'll focus on the major pathway. CBB5 also can break down theophylline, which is missing that 7-methyl group. And the pathway is essentially identical. It uses the same enzymes to pull off the 1 and then the 3-methyl group. And again, you have a major pathway and a minor pathway. And then a few methyl uric acid. These are dead end metabolites. I'm not really going to get into them because the cell doesn't use it after it makes it. And there's not a lot that's produced, and we don't really know exactly why they're produced, but they are there. So we did a lot of studying trying to figure out the pathway, trying to characterize these enzymes that carry out these reactions. And then we also were able to determine the genes that are associated with those enzymes. So we have this ALX gene cluster. This is the gene cluster that's responsible for caffeine degradation. And you see these 5 red arrows. This is the ndmA, B, C, D, and E genes that encode those ND-methylases. When the cell pulls the methyl groups off, it uses oxygen and it actually forms formaldehyde. So the cell needs some way to break down the formaldehyde, and it has those in these 2 green genes you see, the FRMA and B, so that the cell doesn't pickle itself while it's breaking down the caffeine. And then finally, we have these blue arrows you see there. Those are transcriptional regulators that control whether the caffeine genes are being produced, or the enzymes are being produced or not. The black arrows are genes we haven't really characterized yet, but one of them we believe is responsible for helping the cell to transport caffeine through the membrane and into the cell. So those are the genes. We've looked at the enzymes, and I'm going to come back to this pathway map again. As we were working on characterizing this, one thing we thought was, well, is there a way that we could start taking this pathway and putting it into another organism? So say put it into an E. coli cell. E. coli has never been shown to break down caffeine. And so, yeah, it'd be cool to make a strain that can break down caffeine, but even more so, there are some potential applications there as well. So we thought, well, let's just give this a try, and let's just focus on this first step in the pathway where we're going from caffeine to theobromine, and see if we can actually make a strain that will break down, or that will convert caffeine to theobromine, and make a strain of E. coli that will do that. All right, so we set this up where we have a plasmid here. We take the 2 genes that we need. We need ndmA and ndmD in order to carry this out. We put them under control of a single T7 promoter. And this T7 promoter, basically what this means is when the cell makes the RNA from this plasmid, it makes a single RNA strand with both genes on that RNA strand. There is a ribosomal binding site between this RBS. So that's where the ribosome would bind to make separate A and D proteins, but these are encoded on a single RNA strand. Then what we did was we grew up the cells, and then we harvest them, resuspend them in a potassium phosphate buffer, and this is what we're calling a resting cell assay. So the cells are no longer growing. They're just kind of hanging around, you think about they're just sitting around in the hot tub or something. It's not really hot, but they're just kind of swimming around in this phosphate buffer, and we add in some caffeine. So we've grown the cells so that they're making these ndmA and ndmD enzymes. And so they should be able to break down the caffeine. And so we then just take samples periodically and we go to the HPLC to analyze the metabolites. And when we did, so this was our first try, we were able to see, okay, well, we see some caffeine that's being consumed. We see some theobromine that's being produced. That's exactly what we were expecting or hoping to see. Conversion's not the greatest. We're getting about 35% conversion. But for a first try, we were pretty pleased with what we had here. So this was, as far as we know, this was the first time that anyone had been able to see E. coli breaking down caffeine. All right, so our next thought was, well, can we increase that conversion? Because 35% is not the greatest. We'd like to see 100%. So we thought, well, let's take and add more copies of these ndmA and ndmD genes. We had a second plasmid we could do this with, where it had 2 separate multiple cloning sites. So now we have two different T7 promoters here upstream of both genes. And so now what we're getting is two separate RNA transcripts, each of them having their own gene on it. Copy number of this plasmid is lower, so there's less copies of this one than the previous one. But now the genes are separated on their own RNA transcripts. Sometimes that can help in E. coli, sometimes it doesn't matter. But we thought we'd give it a try. And we also can use this plasmid in conjunction with the first plasmid that we constructed and tested. So we put the two of them together. That first one was P81. Here we have this DDA. So when we put them together, we just combine the two names. So P81 DDA. And what you'll see here is with this two plasmid strain, we see caffeine completely converted within 5 hours. In fact, it's probably a little bit faster than that, but our 5-hour time point, caffeine was completely consumed, and we had almost 100% conversion to theobromine. We actually saw a little bit of paraxanthine, which is that minor pathway I mentioned, and that was rather interesting. We weren't quite sure why we were seeing that, but most of the product was theobromine. There wasn't a whole lot of paraxanthine we could do anything with at this point. But it was being produced a little bit. So next thought was, okay, well, we've got this. Why are we seeing so much better conversion than before? And so I've got the first two strains plotted here on this graph, where you see the first strain was in the blue, the second strain here is in the green. We started varying the gene dosage of the ndmA and the ndmD to see what would happen. So first thought was, okay, ndmA, that's the catalytic enzyme. That's the one that's actually pulling the methyl group off. ndmD takes electrons from NADH, passes them on to fuel the reaction. So we thought, well, let's try and add some more of the ndmA enzyme, because that's really what's carrying out the reaction. So we did, on that second plasma, we put 2 copies of A instead of 1 of A and 1 of D. And when we did that, it gives us the red curve here. It really didn't change anything. So all right, well, that's basically telling us we don't need any more ndmA. What gave us that increase was the extra D. So then we said, let's take 2 copies of ndmD and try that. And when we did that, we see a little bit better activity. You know, now we're getting complete conversion in 3 hours instead of 5 hours. So we were able to increase the activity. We got a better strain here, and then went back to see, okay, well, what's the difference here? So what I'm showing here is a protein gel where we separate out the proteins. The first 2 lanes are from the first strain that I showed. They gave us about 35% conversion. The second, or the last 2 lanes are from that best activity strain that we had. S here indicates that we have soluble protein, and for our proteins to be active, they need to be in the soluble form. The insoluble protein is here in the I. So if we get protein here, it's not really functional, at least for the protein we're looking for. So I've highlighted where we see the D and the A. You'll note with our best strain, we have quite a bit of insoluble D. So these are inclusion bodies that can sometimes be produced when you're taking genes from one strain or one species and putting them into a different species of bacteria. Sometimes you get these just because it might be a little more difficult for the cell to fold the proteins properly. So we are seeing quite a bit of inclusion body here, but we also see a pretty good soluble band of ndmD. Whereas with the first strain we had, we don't see very much soluble or insoluble ndmD. There's a little bit there, but it's pretty hard to see. And our thinking is, because we had those 2 genes on the single RNA transcript with this synthetic RBS, ribosomal binding site between, that this RBS that we were using wasn't very strong. And we've actually gone back and kind of used an RBS calculator that's available on the on the internet. And it turns out it wasn't a very good RBS that we used there in the first place. So if we had a stronger ribosomal binding site here, we probably would have had some better activity. That was kind of a post-mortem analysis at this point, just because we had such a better strain with the P81DDD. And we actually used that last strain then to scale this up. Here you see where we've got different concentrations of cells in our resting cell reaction. Not surprisingly, the highest concentration here, we had 20 mg per mil, degraded the caffeine the fastest, produced the theobromine the fastest. We saw trace amounts of paraxanthine, but it wasn't enough to write home about. We were able to take the theobromine, isolate that, and purify it out. And you can see there's a little bit of that here in this vial. LCMS gave us a very pure peak. We tried NMR as well, and it matched perfectly with the standard. So we were able then to take the caffeine, convert it to theobromine using the E. coli cells. And so again, that's this first pathway up here. One thing we've noticed working through all this, though, is the cells are very, or that these compounds are very expensive. Some of them are, at least. So you'll note the red ones here, and in fact, to make this a little easier, these gold ones that I have highlighted here, the prices on these per gram are in the hundreds to thousands of dollars. Whereas the blue ones that we can use as substrates, not very expensive. These prices were taken from Sigma, so these are retail prices, and not really the amount you'd be able to make if you're producing these. But this first step, we weren't really doing much in terms of improving the value of the product. But we have this these same cells that we could look at going from, say, theophylline to 3-methylxanthine, and that's a nice increase in value here. So we focused then on this with those same strains that we had. Okay, so these were the same strains, and the graph looks fairly similar here. We see these, the first couple strains didn't work out that great. This is our control up here at the top where there's no activity, background activity. And in the end, that P81DDD was the best of those strains we tested. Not surprising. But then we started looking at the ratio of ndmD to ndmA, and it really didn't change a whole lot. There's a little variation here, but not a lot. And so we thought, well, let's try and change that a little more, increase that ratio a little bit more. And so we made a couple of new strains. Not even go into all the details. Just note that down here we have this strain that has about a 4-to-1 ratio of ndmD to ndmA. And that turned out to be our best activity strain. In fact, we were able to get complete conversion of theophylline here in 90 minutes. And again, we see about 83% of the product in 3-methylxanthine, about 17% in the 1-methylxanthine. We were expecting to see essentially 100% 3-methylxanthine. So we're still seeing some promiscuity of the enzyme towards the 3-methyl group, and it was actually higher with theophylline than it was for caffeine. But 1-methylxanthine is also very valuable. In fact, it's more expensive than the 3-methylxanthine. In the end, we decided to go with it and see if we could produce these compounds. So we optimized conditions. I'm not going to spend a lot of time on here. Just note we've got different cell concentrations, and we're looking at theophylline being consumed, 3-methylxanthine, and 1-methylxanthine being produced. And as I'll skip these two because I have everything here on this slide where we just are looking at 1, 2, and 4 molar starting theophylline concentration or millimolar concentration. And just trying to optimize the conditions so we could scale this up. You see the higher cell concentration, the more, or the better the degradation, the faster the degradation or consumption, and the quicker the product formation. In the end, we ended up going with this 15 mg per mil at 1 millimolar theophylline to scale this up, just because we could grow up enough cells for that without taxing ourselves too much, and it was still giving us a very good conversion. So we did that. We basically grew the cells up, ran our resting cell assay, added theophylline, and you see here the prep HPLC that we used to separate. We can separate the two products, freeze dry those, and characterize. Now, the 1-methylxanthine, there wasn't enough after freeze drying to do anything with. We couldn't really even scrape it off the freeze drying tray, but there was enough 3-methylxanthine. So we were able to characterize that. And so here's the NMR. On the left is our sample. The right is the Sigma standard. They match. So we were happy with that. And then LCMS looked pure as well. And again, you see that little vial with the 3-methylxanthine in the bottle. So we were able then to carry this reaction out. We were really excited about that. Had a good paper on that process. You know, there's lots of other things we could look at here. One would be going from caffeine all the way to 7-methylxanthine, where we already have the caffeine, the theobromine. What if we just add in ndmB and try to get that? So we've tried that. Doesn't work that great. We get about 30% conversion. We've tried 2 different strains, about 30% conversion, but it's not that great going from caffeine. But if we start with theobromine and use strains that have just ndmBND, you see we can get 100% conversion. And I know I'm going through these kind of quickly here, just trying to show that there's a lot of ways to get to several of these different compounds. The one that I haven't really touched on yet, though, is this caffeine to paraxanthine. And you note the value there of paraxanthine. That would be a really nice process if we could develop to produce paraxanthine, hopefully drive the price down, and maybe make a little money on the side as well. So as we're looking at paraxanthine, so this is actually the major caffeine metabolite in humans. About 80% of the caffeine we consume is converted to paraxanthine, and that's through the CYP, the cytochrome P450 enzymes. In terms of the applications, it could also be used as a stimulant, kind of like caffeine, but there are fewer negative side effects. There are some papers that say it could be used to treat chronic liver disease. It could be a possible neuroprotective agent against Parkinson's disease as well. It's even used in a cosmetic preparation that's marketed in France as a natural skincare and slimming agent. But there's several challenges to producing this. The chemical synthesis is really difficult and expensive, which is why paraxanthine is so expensive. There's limited natural sources. Plants don't make it. Bacteria make very little. Humans make it, but we can't really go around extracting paraxanthine from humans. So we're stuck with the chemical synthesis. We've tried taking the human enzymes and expressing those in microbial cells. They just don't work very well in the microbes. So we went back to look at these enzymes we have. And for the longest time, we've been saying, well, we have caffeine is converted theobromine by ndmA and to paraxanthine by ndmB, because ndmB is the one that pulls off that 3-methyl group. As we go back and look at the kinetics of these 2 enzymes, and I know there's a lot on this slide as well, but let me just point out here, you focus on this kcat over km value. That's a measure of enzyme efficiency. So the higher the value, the better this enzyme is at running the reaction. You note for ndmA with caffeine, we're about 5.1. ndmB is 3 orders of magnitude lower when it comes to caffeine. So ndmB really just does not like caffeine, whereas ndmA really likes caffeine. And ultimately, ndmA was evolved to break down the caffeine, and then ndmB was then evolved to take the theobromine that was generated from the caffeine and convert it to 7-methylxanthine. So that N1-methyl group that's here on the caffeine tends to block the activity. We see that because theophylline is also a pretty poor substrate for ndmB. So the compounds with the 1-methyl group are not very good with ndmB. Pull that off and it works pretty well. Well, we went back to look at some homology modeling and try and figure out why. And so what I'm showing here is the binding pocket of ndmA and ndmB. And these are just some homology models. It's not actual crystal structure. But it indicated that this salmon residue right here, this is a tryptophan. It's sticking out in the binding pocket. So here's the caffeine in the yellow and blue. And this tryptophan might be sticking out in the binding pocket, reducing the volume of the binding pocket. So there might be some steric hindrances just because of that N1-methyl group. We actually tried taking this and putting the B with the D into the cells, and there was really just no activity towards the caffeine. We thought maybe we could get a little paraxanthine. There was nothing. But again, when we had these ndmA cells, and I kind of showed this before, you know, we go from caffeine, this is our HPLC chromatogram at 1 minute. Here we have the caffeine peak, to 30 minutes, caffeine's mostly gone. We get mostly theobromine, but there's that little bit of paraxanthine. And so again, just plotting the concentrations, a little bit of paraxanthine, not a whole lot, mostly theobromine. So one thought we had was, and I'll back up here, one thought we had was, well, ndmA is really good at this, at breaking down the caffeine, but it really likes to pull off that 1-methyl group. Could we maybe engineer this to pull off the 3-methyl group? So we actually tried to do this. We tried some different mutation experiments, just with some random mutagenesis to see if we could get something. But the screening process is really time intensive. It's just really difficult to do. We could never get a good hit. That's when we were actually blessed enough to have someone reach out to us, a professor at Korea University who reached out to us to say, I've got a student that wants to try and crystallize your enzymes. I'd been trying to find someone around here to crystallize the enzymes, and no one would return my emails. So I got really excited and we started working together. I was even able to send a graduate student over to Korea for the summer to kind of work on this as well. And so they were able to get some crystals. Here's a crystal of ndmB, but they were able to get crystal structures of the A, the B, and also the A with caffeine. And so you can see the A and the B are kind of overlapped here. They look fairly similar. There's a couple differences. And when they look at the enzyme, the residues in the binding pocket that align that ndmA and ndmB, or sorry, that align the caffeine in there, they found that there are about 9 amino acids that really play a role in trying to get the caffeine lined up just right for the reaction to occur. Of those 9, 7 match up with ndmB, but there's 2 here, this asparagine and phenylalanine, that are different. In ndmB, it's a glutamine and a leucine. So the thought was, well, let's, what if we were to mutate these 2 residues in ndmA so they would match those in ndmB, because that's good at pulling the N3-methyl group off. It just doesn't like the caffeine. So we gave it a try. So we tried we tried a single mutation here, the N282Q, and you see after 60 minutes, there were a couple little products, but not much at all. The second mutation we did, still see mostly theobromine. The rate was a lot slower. So this is after 2 hours, whereas with the wild type, we'd see most of the caffeine gone within 30 minutes. But again, most of the product's theobromine. So neither of these was looking that great, but we also tried the double mutant just to see what it would give us. And what we see here now, for the first time, you see the paraxanthine. We actually had a fairly decent peak of paraxanthine. You see some theobromine as well. The peak's a lot smaller. The rate's still really slow. You see, we still have all of this caffeine after 2 hours, but now we're actually getting some paraxanthine. And so if we plot out the data, I've got them all here, but for the first mutant, we did see some increase in paraxanthine in the product pool, up to about 28%, but with the double mutant, now we're seeing 85% of the product that's formed from caffeine is paraxanthine. So, you know, after we finish jumping up and down and clapping our hands and getting all excited, we thought, okay, well, that's great, but the rate's still really low. We go back to the crystal structures and look, and here we have ndmA in the green, ndmB in magenta. There's these 2 variable loop regions that don't match up very well. The A here is the yellow. It doesn't match that great with the ndmB loop. But there's this other orange loop that in ndmA, when caffeine binds, this actually closes down on the binding pocket and kind of tucks the caffeine in there. And we don't really see that with the ndmB structure because this didn't have a substrate in it at all. So we thought, what if we take this this loop structure from ndmB and swap that in place of the ndmA loop. So hypothesis was that we swap these loop structures, that would help increase the activity of our double mutant. So we did that, and I'm showing the HPLC chromatograms here from the double mutant with the loop mutation, 2 different cell concentrations, so an OD of 10 and OD of 50, and look at this paraxanthine peak right here. So that's the best paraxanthine peak we'd seen. Caffeine's being consumed, we're getting paraxanthine. Again, it's not super fast, but it's at least getting better. So here I've plotted different cell concentrations. Not surprisingly, the higher the cell concentration, the faster the reaction, and the more caffeine consumed and the more paraxanthine produced. And so with our highest cell concentration, you see here we're consuming caffeine. Our paraxanthine's plateauing off at a little over 400 micromolar. That's been fairly consistent with this batch of cells. And so we're trying to work around that now. We've got some promising leads that we don't have all the data on it, but I think as we improve our redox recycling inside of the cells, that we'll be able to increase that as well. We have some preliminary results that show that by regenerating the NADH, that improves the conversion. I just don't have those plotted and ready to show here yet. So kind of in conclusion, what I'm showing here is we're able to take using the crystal structures, we were able to demonstrate that we have those 2 residues that were important, and with the loop mutation, we can get up to about 40% paraxanthine. So we're really excited about that. We're working on, we've got 1 paper that's in revision right now. We've got another one we're about to submit on this paraxanthine production. And so that, this has kind of been one chapter of the research we've been doing in my lab. It's definitely the most productive, and we're really excited about where it's going. I'm going to skip over these last couple of slides that we're just talking about the modular plasmids that we were doing. But basically, this is a project that we were doing with our iGEM team to just make some really cool plasmids that we could use, and you can swap out these modules fairly easily. So I just want to kind of conclude here and just thank everyone that's worked on this. I don't get to go in the lab very often, so I'm grateful to the students that do all the work. I currently have 2 PhD students that are working on this, former graduate students. So Shelby Brooks just finished her PhD this last May, and so she did a lot of that paraxanthine work that I presented. These are the current undergraduates in my lab. I've had several others that have graduated as well, and then also funding sources that have given us money so we can do the work. So I'm happy to take any questions. I don't think I saw any in the chat come through. So if you guys have any questions, I'm more than happy to answer for you.
Fantastic. Thank you for that presentation. So you can either ask a question by unmuting your microphone, or you can put it in the chat window. So we're going to open up some time right now for some questions. So I just had a question maybe to start with. So the crystallization of your enzymes, I imagine that, to have another group kind of recognize what you're doing and then want to extend things a little bit more, that's pretty exciting when, somebody takes hold of it. Can you say anything about, do you feel like you can get better selectivity? There's been a lot of research like on microreactors or channel reactors or things like that, like either in different phases, different pressures, things like that. Like, are there ways that you feel like you could improve the selectivity, or you had good selectivity for some of those, but the rates were really low? Yeah. You feel like you can, any insight on that? Like, what would you, if you had another group approach you and say, hey, we see a lot of promise in this, we'd just like to be able to help you with some of the rates, what direction would you go on that? What would you recommend?
Yeah, that's a good question. A lot of what we've focused on for the rate has been more trying to improve the enzyme, right? Or improve the strain. I think there could be some reactor design approaches that are out there, whether, like you said, some microreactors or something. I don't know if maybe doing some cell immobilization or something would work as well. to try and improve recycling. So those are some of the first things that jump out at me. You know, like I said, most of what we've done is focused more on trying to improve the proteins themselves to just give us better activity. But eventually we're going to get to a point where we just can't improve anymore. And you're kind of limited by the thermodynamics there of the proteins. So yeah, I think definitely some reactor design approaches, whether that be looking at some sort of immobilized cell system with a plug flow type reactor, or some other approaches there would definitely be beneficial.
Okay, we had a question in the chat window.
Yeah,
more
so
about
Ken,
students working remotely. So what do you think about that?
right. So Ken, so yeah, did the student I sent to Korea already speak Korean? No, she did not. She really enjoyed it. The lab she was in spoke English when she was there, and she really, really enjoyed it. So that's a good question. And Andrew, your question on scalability of the process, I forgot to mention that as I was going through this. Let me see if I can pull that up here. This was a slide someone at Iowa had developed once, talking about the bacterial cafe. I thought was kind of funny. Coming back to that 3-methylxanthine production that I talked about. So we grow the cells. We were able to grow about 4 liters of cells and have about a 1 liter resting cell assay. So in terms of scale, it's still very much bench scale. I mean, I'm sure it could be scaled up quite a bit, but you know, we're just producing milligram quantities of the product at this point. Let's see. Any interest from industry, perhaps pharmaceutical companies? There has been some interest. Well, let me back up. There was some interest in the paraxanthine, and we were excited, but that was before we had all of our breakthroughs. Since then, we haven't been able to get the company to get back in contact with us. So hopefully, we'll be able to get some more interest in the future with some of this, because I think it's really cool. And I know there's been some interest in the past.
Okay, any other questions? Okay, well, I think we're about out of time right now, but this has been a fantastic presentation. I really appreciate you sharing your research. You know, it's interesting to see how you know, catalysis. I mean, this is really just a biocatalytic processes and reaction mechanisms. I know a lot of students are working on those, trying to understand fundamentally what's happening, whether that be with, you know, forest fire combustion or other types, you know, that are happening at BYU. So it's interesting to see the approaches you're taking and the mechanisms and pathways that you hypothesize and use experiments to confirm those, or to, you know, to look for other interesting or useful ways to process these chemicals. I just had one final kind of question about this. You've kind of focused on you know, the process of creating high value chemicals, but I imagine that there's also a lot of interest in neutralizing waste from food processing as well, and there's probably a lot of value in that. Can you say, I guess, anything about the potential industrial application of cleaning up waste versus like creating high value chemicals?
Yeah, that's a good question. That's something we've looked at a little bit. In terms of, you think of these coffee and tea processing plants, for example, or even, you know, a lot of these are going to be found in, you know, other countries where they grow the coffee, for example. They have all of this waste that's generated, and a lot of it still has caffeine in it. A lot of the plant material still has caffeine. And so that caffeine can get out into the soil and into the water. It's toxic to most insects and bacteria. So they try to do a lot of composting. They've looked at using it for animal feed, but the animals don't really like it that much, partly because of the caffeine as well. So we have looked at potentially taking this as well and trying to look at, we'd make a yeast cell that could break down the caffeine to xanthine or even further. And then you now have material that could be used as animal feed, or it could be used as fertilizer or something like that. So we haven't really done a lot with that, but that's definitely been something we've been interested in.
Okay, fantastic. Well, thanks, Ryan. This has been a really fascinating presentation. I really appreciate that. And thanks everybody for joining and for putting some things into the presentation. And don't forget to connect with Dr. Summers. I put the LinkedIn profile there, and don't forget also to record your attendance on Learning Suite. So thank you so much, Dr. Summers, and we'll talk to you later.
Thank you. I appreciate the opportunity.